It's my great pleasure to introduce our first keynote speaker. Thom Sharp joins us from the University of Bristol, and we're really starting on the not a very small end of the resolution scale here with in situ structural immunology, where we'll be looking at the smallest perspectives on the immunology landscape, and really looking forward to hearing about how protein structures in whole cells can get determined in the immunology context. Thom, please take it away.
Thank you very much. Can you see my screen and hear me okay?
Yes, that's perfect.
Perfect, great. So hi, good afternoon, everyone. It's a real pleasure to present to you all today. And I'd also like to thank the organisers and, excuse me, there's always a problem at the start. And of course, Communist Globe for the opportunity to present. So my lab in Bristol is quite new, moved here at the start of this year. We combined structural and synthetic biology to understand and control the human immune system. And I'm going to tell you today about how we can use cryo-Electron tomography to image immune system components such as these, caught in the act of immune defense, and how these structures can be used for drug design. And one of the things we learned along the way is that every time we move towards a more native-like system, we learn more about how our immune system is activated. And so I thought I'd end by talking briefly about some of our recent developments to make in situ structure biology routine, how we can solve the structure proteins in their native environments. So my lab is interested in the human immune system, multiple aspects of it, but today I'll focus on part of our immune system called complement. And complement plays a key role in immune defense against invading pathogens, as well as clearance of cellular debris, and this helps to protect us against autoimmune diseases. So complement components circulate in our blood and tissues in an inactive form. And the classical cascade, which I'm going to talk about today, can be activated when certain antibodies bind to antigens, and this might be on a bacterial cell surface. And when they bind, they form this platform. And this platform acts like a landing pad for the first component of complement, called the C1 complex. Now, C1 comprises this ligand binding and scaffolding complex called C1q in blue, and two serine proteases, C1r and S in pink and purple here, that form a heterotetramer inside of C1q. Now, the classical cascade can also be activated by proteins called pentraxins. These are kind of like the innate immune system's versions of antibodies. These are fascinating and largely overlooked proteins. They've got roles in immune defense, synaptic trimming, female fertility. They're now being associated with diseases like COVID, flu, Alzheimer's, but sadly I don't have time to talk about those today. But whether it's initiated by antibodies or by pentraxins, complement activation by the C1 complex is quick and efficient. Binding of blue C1q induces activation of C1r and S, and this initiates a cascade of events, resulting in opsonization of, for instance, the bacterial membrane. And what that means is the membrane is covered in proteins that act like seasoning for macrophages and neutrophils. They want to come along and phagocytose or eat this cell. And while the macrophages are on their way, complement is busy destroying the bacterium itself. The cascade terminates with the formation of a pore called the membrane attack complex, which lyses the bacterium. So if it's not been eaten by macrophages, it will be popped by this mac pore. Now, we've spent a lot of time studying antibody-mediated activation, indeed solved the first structures of these initiating complexes with IgG1 and IgM, as well as the terminal mac pore. Last year, we actually imaged IgG3-mediated activation, which I'm going to talk about today. And this year, we managed to solve the first pentraxin-mediated complement complex. Now, before I talk about the results, most of these structures, all of them with the grey outline, have been determined in situ, in their native environment, which is human serum. So I thought I'd briefly talk about how we actually did this. How do we collect these kind of in situ data? Well, we use a technique called cryo-Electron tomography to get these 3D data. Cryo-Electron tomograms are collected in much the same way as an MRI or a CT scan, in that the same region is imaged from multiple angles. And this gives us a series of tilt images. So here's one such tilt image. This is a liposome, a kind of mini cell mimetic, if you will. It's about 100 nanometers across, embedded in vitreous ice, and the whole thing is suspended between these dark carbon threads. Now, all these little black dots are gold beads that we can track with very high precision to give us a high resolution alignment of the tilt series. And when we have the aligned tilt series, we can computationally back project them to yield a 3D volume. And now we have this volume, we can slice through it in any direction. Here, I'm just going up and down through the volume. But this allows us to see the individual complexes much more clearly. So on the outside of this liposome, we've actually got a load of IgG1 antibodies bound. We encapsulated a bunch of enzymes inside that we're using as biophysical readouts. And you can see all they're related to each other in 3 dimensions. Now, with these volumes, we can take these 3D tomograms and cut out and align small sub-volumes or sub-tomograms containing our particle of interest. Here, there's one of those MAC pores lysing this liposome. We can extract it, along with many, many other instances of this MAC pore, align them and average them, go through many iterations of this, and we end up with a much higher resolution map that we can interpret with de novo models, crystal structure fragments, AlphaFold, you name it, to give us so we can interpret these to much higher resolution. And when we have those higher resolution pieces of data, we can actually place them back into the tomogram. And this is how we can interpret atomic interactions in situ. Someone coined the term molecular sociology because you get to see how these things are related to each other in 3D. And this is the basis of our aim to do what I call in situ structural immunology. So I'll briefly tell you about how we applied this in situ structural immunology to IgG3. This is the strangest, it's a weird IgG subclass. Just as a bit of a reminder, we have 4 subclasses of IgG antibodies floating around our blood, named after their abundance in sera. And these have different but similar structures, but they activate complement to widely different degrees. So IgG3 is the best, closely followed by IgG1. IgG2 is all right at complement activation. IgG4 is terrible at it. Now, the structure of antibodies that you'll all be most familiar with is IgG1. This is the typical gamma shape or Y shape of antibodies. We have these 2 Fab domains that bind to the antigens, linked to an Fc region, which is the kind of effector function. And these are linked with a short hinge region, which has disulfide bonds between them. So that's IgG1. IgG3, in contrast, has this uniquely long hinge. It's got 11 disulfide bonds, 6 O-link glycosylation sites. And this means that IgG3 is the most flexible subclass. Now, most therapeutics, something like 95% of them, are based on IgG1, and some use IgG2 or IgG4, but there are currently, at least to my knowledge, no therapeutics based on IgG3. There's also no high-resolution structural knowledge of IgG3, and these 2 points are linked. It's hard to exploit something when you don't even know what it looks like. So we decided to solve that. This was work led by Leonie Abenstein in my lab. She just moved to Vienna to take a postdoc up there. So we were making these cell mimetics. These are antigenic liposomes. We can make them the perfect size for cryo-electron tomography, which is about 100 nanometers across. But we can also use them as readouts for complement activation. So by encapsulating a very high concentration of dye called sulforodimine B, this is a self-quenching dye, so it doesn't fluoresce. We have antigens on the outside of these membranes. When we add antibodies and human serum, we get complement activation, MAC pore formation. The pore lyses the liposomes, it allows the dye to leak out, and then we get an increase in fluorescence. And that's what you can see here. So this increase in fluorescence is caused by, in green, IgG3 mediated complement activation, and in red, IgG1 mediated. So what you can see is here is IgG3 is better than IgG1, which is what I told you about earlier. Now, what's nice about these liposomes is that we can take them at any time point of this curve. So before we add the human serum, right at the end, halfway through, wherever we like, and prepare them directly for cryo-electron tomography to see what's going on at a specific time point. So here I've incubated, or Leonie rather, incubated these liposomes with IgG1 antibodies. And we're going to slice up and down through this tomogram of a liposome. And on top of the membrane are all these little antibodies. So this is a patch of antibodies stuck on the outside of the liposome. That's what we've seen a bunch of times in a few different papers. Here's IgG3. It's the first time it's been seen. There's a big difference. Now you can see these striations that are perpendicular to the lipid membrane. This movie's a little bit jumpy for me, so I hope you can see it clearly. If you can't, I've got some stills because I was worried that would happen. But here you can see that here's the liposome, and we have these beautiful striations kind of sticking out of the surface. We'd never seen these before with IgG1. So these striations are 7 nanometers tall. And as I told you a few moments ago, we can align and average images like these to get us a better insight into their structure. So they're 7 nanometers tall, and they're also 3.8 nanometers wide. Now, these slices through the 3D tomograms are actually of the kind of equator of the liposome. So we're slicing through right in the middle of our sphere. But we can actually see all of it. So here we're going to go from the South Pole through the equator up to the North Pole. So there's the equator. Hopefully it's playing for you. And then right at the North Pole, we see these other slices. Again, just in case the movie didn't play, doing things online is always a bit tricky for movies. This just tells you kind of where we are. So before we're looking at the equator of the liposomes, where we see all the striations, we're now looking at the North Pole, kind of like the nadir or the apex. And now if we zoom in here, we can see these striations. These are actually 5.25 nanometers apart, not the 3.8 we saw before. But again, we can align them and average them. And when we do that, we see this beautiful 2D array appear. So here we can actually take a Fourier transform. We can see that these stripes are separated by 5.25 nanometers in the vertical direction and 3.8 nanometers in these horizontal stripes. And the 3.8 nanometers actually come in pairs. But this gave us a lot of information about how these things are separated out. So we have 3.8 nanometers in one direction, 5.25 nanometers in the other direction. If we take a look at a fab domain, these values are suspiciously similar. So what we're actually looking at is a 2D array of highly ordered fab domains. These are on a slightly skewed lattice, separated by 3.8 nanometers in this direction and 5.25 nanometers in this direction. So we could build a theoretical model that explain these data. Now here, the light and the heavy chains are colored yellow and blue respectively, but this is arbitrary. We don't have the resolution to tell. So here I've got it going light, heavy, but it could be going light, heavy, or it could be a mixture. We need higher resolution to tell. We also can't tell which two fabs belong to one antibody. So each antibody has two fabs. I've colored here two blobs. This might belong to one FC, it might be these two, or it might be these two. Again, without high resolution, we don't know. But the amazing thing is we've only ever seen this for IgG3. And of course, we're trying to get high resolution information right now to improve these maps. But back to these slices, some of you may have also noticed that there's this little line above the striations just here. Now, we'd seen this before. This is 22 nanometers above the surface. So we'd seen these before in IgG1 liposomes. And they usually corresponded to the FC parts of the antibody, actually forming a hexameric FC platform attached to the fabs. But now this platform is 15 nanometers above these striations. So again, we picked a few thousands of these particles and performed subatomic averaging and found they were indeed hexameric IgG3 FC platforms. So this is the effector domain of antibodies actually form these beautiful hexagonal structures. Now, IgG3, of course, has this much longer hinge I've already mentioned, and that's what's elevating this platform 22 nanometers above the surface, above the membrane. Now, we could combine our models of the FC platform and the fab array to yield a complete model of IgG3. Now, this extended hinge separates the FC and the fab domains by 15 nanometers. We couldn't determine the structure of the hinge itself, so we have this hybrid model instead. I actually don't think the hinge has a single structure per se. I think we would have found it if it did. So I suspect, as other people have previously shown, that it's a semi-rigid but flexible polypeptide, and that's how we model it in here. Now, of course, we didn't stop there. We could then add human serum to look at the C1 complexes on top of these antibodies. That's what you can see here. On the left is IgG1, on the right is IgG3. We've done it on IgG1 a bunch of different times, so we knew exactly what to look for. But again, we see these C1 complexes sometimes above these striations, sometimes not. We could see these C1 complexes, extract and align them, and give us a 3D map of antigen-bound IgG3 C1. The whole map was quite low resolution, but we could actually zoom in different parts and get much higher resolution. And this allowed us to build a model. So we have this elevated hexameric IgG3 FC platform in green, kind of in the middle here, with the fab array at the bottom. On top of that is C1q in blue that binds to the antibodies. C1r is this purple protein that sticks out kind of parallel to the membrane. And C1s, the serine protease domain, bends down to this orange colored protein. This is C4b. Now, again, I'd just like to remind you, this is all done in unpurified human serum. Everything that's in your blood, except the cells and the fragments, are still there. C4b is the product of this enzyme complex. So by using cryo-Electron tomography in an unpurified sample, we're actually able to catch this enzyme in the act of initiating immune defense. Now, from these structures, we learned loads about how IgG3 functions as an immune system activator. We could hypothesize a load of reasons about how IgG3 is just the best antibody. It's best at pathogen neutralization, antibody-dependent phagocytosis, and complement activation. I'm not going to talk about those things today, but you can go away and read them in our NatCom paper from last year, if you like, or drop me questions. Nice thing about doing these things online is even if we don't have questions at the time for questions at the end, please just type a message to me and I'll try and respond to as many as possible. But instead, I wanted to show you how else we can use these structures. So by solving these kind of structures, these similar but different structures of IgG1, IgG3, and IgM antibody complexes caught in the act of initiating immune defense, we could see what changed between their solution phase, their inactive complex, as they're floating around our blood. And what's common between all of them in their antigen-bound active state, in their antigen-bound active structure. And we discovered actually that the binding interfaces between C1q and the antibodies themselves are actually tiny peptidic domains on the periphery of these FC platforms. So we've got this whole antibody complex. The only bit that C1q binds to is right on the edge of this FC platform. So we had an idea. We designed and synthesized a peptide comprising just these amino acids. This is what it looks like in our structure. So this is an IgM C1q complex. We could then cyclize this little region and kind of enforce it to look like it looks like in the whole antibody structure. This was work done by my PhD student, Sebastian Hammers, and a collaborator called Amy Boyle. So we could take this peptide, and when we conjugated it to a lipid and added human serum, we found that the peptidic interfaces on their own were sufficient to activate complement. So these peptides are 1 kilodalton in mass compared to the 950 kilodaltons of the IgM complex. So what we've been able to do is actually distill the complement activating ability of an antibody into something 0.1% the mass of an antibody. So we had to come up with a catchy name for this. So we called them peptactins for peptide-based activators. Now these peptactins weren't very good. I've written here first generation peptactins. They weren't very good, but they showed us that the concept worked. So what we did next was some structure-guided selection and design. We used a technique called rapid selection, which we did in Senyo Jonke's lab in Amsterdam. And this allowed us to evolve our minimal motif, this little peptide, into a more potent molecule. And now we have second generation peptactins that can sequester or recruit C1 from human serum selectively. It can activate the C1R and C1s serine proteases, and it even induces pore formation and lysis of lipid membranes. So what this kind of means is that we've taken an antibody and turned it into a drug-like small molecule just for complement activation. There's loads of other things that antibodies done, we've ignored that, but the complement activating ability of antibodies, we've managed to distill into a peptide around 16 amino acids long. So I call these drug-like molecules. So what are we going to do with them? Well, these are little things that we can stick on almost anything and give them the ability to activate complement. So here we stuck this tiny peptide on a lipid, and it can activate complement pretty much as effectively as I'm getting some feedback. I don't know if someone's got there. Perfect. They can activate complement just as well as an antibody. So we're now in the process of sticking them on various other things that don't have the ability to activate complement, different targeting complexes. So Fabs on their own, for instance, can't activate complement. We can give them that ability. Nanobodies famously don't have any effector function. They're just binders. We can give them ability to activate complement. So we can get smaller and smaller in these things all the way down to targeting peptides. So we're exploring things like, can we treat antimicrobial resistant bacteria? Can we kill B cells? So these are dividing uncontrollably in lymphomas and leukemias. If we can kill them, we might be able to use our own immune system to destroy these cancer cells. B cells also secrete antibodies erroneously sometimes to other parts of our body, and that's how you get some autoimmune diseases. So we're looking into, can we target these to specific B cell populations in order to remove only those B cells that cause the disease, leaving the rest intact. So we've got some ideas and we're working through these now, but the point is all of these were made possible only when we imaged these molecules in their native state. And so for the last 5-10 minutes or so, I'm sure someone will yell if I start to overrun, I thought I'd talk about how we're moving towards doing it all in situ. So hopefully I've convinced you that imaging proteins in the native environment is essential for us to learn more about how complexes actually function. So obviously we want to move towards the most native type of systems we use, whole cells. So I'm going to talk now about some work done by Martin Dotel and Mart Last. Martin's currently in my lab. Martin's now a postdoc at a Max Planck Institute. We've got a few publications on this. Mart has actually just written a couple of book chapters explaining how we do this, basically. Not quite an idiot's guide, but it really talks through step by step about how we do all these things. But first, I'll put this into context. So a single one of our cells is around 20 microns wide, whereas an E. coli bacterium is kind of 2 microns wide. It's pretty much the smallest organism we can see with traditional light microscopy. Viruses are 200 times smaller than our cells. You can't see them with light microscopy. We need electron microscopy to first see them. And most proteins are another 10 times smaller than that. So a single antibody protein is around 2,000 times smaller than our cells. Now this movie is to scale, so you can see that proteins are tiny. But with EM, we can see them. Cryo-EM lets us see their constituent atoms. So now we have a different problem. Even though we can see them, how do we find these tiny proteins? Well, by using biochemistry, we can add a fluorescent protein to our molecule of interest, guiding us where to look. This, of course, is one of the major advantages of light microscopy, and we can combine that with electron microscopy to perform correlative light and electron microscopy, or CLEM. We and a few other groups have pioneered several methods to do CLEM and cryogenic temperatures, so-called cryo-CLEM. And most recently, my lab has decided to combine this with super resolution cryo-light microscopy to perform super resolution cryo-CLEM, or SR cryo-CLEM. And this allows us to find proteins with 30 nanometer precision. Now, there's a few methods to perform super res light microscopy. The most applicable for cryo-CLEM is single molecule localization microscopy. This includes techniques like Palm and Storm, and generally delivers the highest resolution. So here's what our sample might look in a conventional light microscope. To do single particle localization microscopy, we can activate only a small subset of these spatially separated fluorophores. And if it's just one fluorophore on its own, we can find its center with high accuracy and plot it. We can then turn that fluorophore off and turn on another fluorophore, plot its center. And as long as the fluorophores are spatially separated, we can build up these super resolved reconstructions to give us much higher resolution. Now, VIDI was the Dutch Research Council grant that awarded me some money to do a lot of this research that I'm presenting in this part of the presentation. Now, perhaps unsurprisingly, we very quickly found out that photons devitrify the ice. And this completely ruins cryo-imaging. So we found that devitrification is dominated by the laser intensity and not the total dose or the duration of imaging. This is actually the opposite to cryo-EM, where it's the total dose that's the problem. Here, it's just the dose rate or the fluence. So in fact, what we could do is avoid devitrification entirely as long as we stayed below a certain threshold. So here it says 30 minutes. We've done this for up to an hour, and we see no devitrification below a certain laser power. If we increase this just a little bit, within 5 minutes, it's actually pretty much instantaneous. We start to see this cubic and hexagonal ice formation. This is devitrification of our sample, completely ruins cryo-EM. This was a consistent phenomenon, with devitrification starting in the center of the grid squares and moving outwards from there. But this was dependent on the kind of grid you use. We actually came up with quite an elaborate mathematical model to explain all this, but in the end went down the pragmatic route. We basically bought as many different grid types as possible that are commonly available and tested them. And that's what you can see here. So we published this last year. The best ones we found were ultrafoils. So these are gold grids that we can grow cells on with a gold film. And for that, we could pretty much triple the laser intensity we could put on, or more than triple, really. So they're the ones we use now, these ultrafoils. There were a couple, form var film, which is kind of plastic, and silicon monoxide, which is glass. We couldn't devitrify things on them. So that looks like it'd be perfect. But both of those spontaneously blink. And when you're trying to find these individual fluorophores, if your background is also blinking, it's basically impossible to do single molecule localization microscopy. But we are working on that to see if we can get like a perfect support film. Just in case you were wondering, we were wondering as well, just because we don't see devitrification, are we still damaging the proteins? The answer is no. So we can put in 3.6 million photons per square angstrom with no decrease in resolution whatsoever in our proteins. So for fluorescence light microscopy, we need fluorescent beacons. Traditionally, we use fluorescent proteins like GFP, but GFP is not photo-switchable. For super resolution light microscopy, we need photo-switchable proteins that can be turned on and off with different wavelengths of light, such as RSE-GFP2 shown here. And this is what you can see. We're turning it on with a pulse of 405 nanometer light, and then we can actually turn it off again with 488 nanometer light. So we can turn it on and off, and that's how we get those spatially separated fluorophores. With GFP, you see there's basically no photo switching. We discovered a few years ago something rather fascinating. When we prepare fluorescent proteins for cryo-EM, even non-photo switchers like GFP become photo switchable. So we can turn them on and turn them off, just like traditional photo switchable proteins. And this means that they can actually be used for super resolution cryo-CLEM. And this is true of all of the green proteins we tried, including non-switchable GFP, activatable proteins, slow switches, fast switches, reverse switches, positive switches, negative switches, photoconvertible proteins. All of them behave pretty much the same at cryogenic temperatures. And this is exciting because it might mean that you don't have to reclone your protein in order to use this technique. If your fluorescent protein spontaneously starts blinking, you can do super resolution cryo-CLEM. They weren't all the same though. One of them had a much higher dynamic range than the others. This was RSE-GFP2. So we use this to label intracellular microtubules, and that's what you can see here. We tagged a protein called MAP2, a microtubule associated protein. with RSE-GFP2. And this is inside a whole mammalian cell. So the cell is much, much larger than I've shown here. But if we zoom in here, we can see the cryo diffraction limited light microscopy image. And we can compare this with the super resolved reconstruction. And this really shows you the power of this technique. We can also correlate this super resolution light microscopy image with three-dimensional cryo-EM on the same sample. And this we can then combine with structure determination using subatomic averaging to solve structures of proteins in situ, in our cells. Now in Bristol, we've just, I wrote a grant the first month I was in Bristol actually to get a cryo-FibSem. And this is going to allow us to thin out pretty much any sample that we can prepare for cryo-EM. We can now make perfectly thin enough for cryo-ET. So we should be able to get almost any biological sample and do in situ structural biology. So we've actually also written some software called SC Nodes, which automates all the data processing that I've shown you here in a very, or at least it automates a lot of it, but it's very intuitive. It's a lot of, every time you change a parameter, you can see how it affects your reconstruction, your correlation. You can pick particles from it. You can do some basic in situ structural biology in that, which you can then take out to more dedicated programs. We've written a few book chapters on how to do this. Everything's open source, so we put everything on GitHub. All our papers are open source. Basically, I want as many people as possible to start using this technique because it really is excellent. And we're now starting to apply it to multiple aspects of the human immune system, which hopefully I'll be able to tell you all about if I'm invited back in the future. So to conclude, I've shown you that cryo-electron tomography provides this functional information by solving in situ structures of proteins caught in the act. IgG3 forms these extended 2D array of Fab domains, and the extended hinge of IgG3 results in the SC portion being a long way from the membrane. I didn't have time to talk about the functional consequences of that, but there's a lot of them. I also showed you how the structure could be taken from a 1 megadalton antibody complex and distilled into a small peptide that's still able to activate complement. And hopefully at the end, I could convince you that structures of intracellular proteins can be solved to sub-nanometer resolution within whole mammalian cells using this super resolution cryoclem. And just so you know, I'm soon going to have 2 postdoc positions open, one to work on antibody engineering and one to work on methods development of this super resolution light microscopy and methods thereof. So if you're interested or have experience with either of those, please do get in touch. The adverts will be up soon. That just leaves me to thank my lab members. I've talked about Leonie Abenstein's work. She did a lot of the IgG3 work. Sebastian Hammers did the drug design, structure-guided drug design, and Mark Last did a lot of the super resolution work. But really, my lab is highly collaborative and cooperative, so I want to thank everyone in my lab, all my wonderful collaborators, the UKRI, the ERC, and the NWO for funding. And of course, thank you all for your attention. I'm very happy to take any questions you have.